All right, so today what we're going to do is we're going to continue discussing the viruses. We are going to, the beginning of next lecture, we'll move into viroids and prions, and the next lecture, about the first 9 slides, will be on the midterm, and then the rest of that lecture will not be on the midterm. So last week we started to visualize the importance of different viruses before we get into, come on, present. Before we get into this week's lecture, and while the projector decides to turn on, what were the major points that we got out of last week's lecture? How do we categorize viruses? What are the important characteristics of viruses that allow us to categorize them into different groups? Anybody? Yep. Okay, so their genome, which can be basically DNA, RNA, single-stranded, double-stranded, positive, negative, right? So that's an important criteria. You said the capsid. So what about the capsid? Its morphology or its shape? Yes. Okay, so what type of capsid it has. What about whether or not it does or doesn't have an envelope? Is that an important criteria? Okay. Let me just see if we can make this projector actually turn on. All right, let's see what's doing this. Okay. On. Now it's off. Such is technology. All right, so we have the DNA, RNA, so the genome, the shape of the capsid, does or doesn't have envelopes. What else can we use as a criteria? Anybody? So we can talk about the disease it causes. Is that the ideal way we can categorize viruses? Is there a problem with categorizing based on a little bit of a problem, using disease as a reason or a way to categorize?
Yeah, because a lot of the symptoms that they create are pretty universal.
Right. So it is useful, but it's not ideal. What do you think about host range? Can we categorize viruses based on host range? For the most part, we can. Most viruses are relatively limited in their host range, in that it could be a plant, an animal, a bacterium, and depending on some viruses, it'll go all the way down to what level. So a virus in plants doesn't just infect all plants, it tends to infect only certain, sorry? In plants, there's a less of a tissue specificity, but there can be some, but the genus level, right? So we have a tobacco mosaic virus. And that's the same with all viruses. For example, we have viruses that infect only E. coli in the bacterial world, et cetera, or other bacteria. We have viruses that have sometimes a more broad ability to infect a number of mammals, but then we have only the viruses that can infect humans or other specific organisms. So we do have some, yep, yes, Andrew.
You were just mentioning the tobacco mosaic virus. Would that be, like, when you say genus, would the tobacco mosaic virus?
Well, it's the genus of tobacco. I'm not going to bother you with the name of the genus of tobacco, but it's the one that, it is tobacco, which is its common name.
Is it the same or different than the cucumber mosaic virus?
It's completely different. Yeah, it's not the same virus. Okay, all right. So what we're going to do today is spend a lot more time talking about viruses that infect mammals, but we're also going to talk about, within the context of that, the different stages of viral infections. So this table is just useful because it tells us some important characteristics of human viruses. So what we have is the names of the viruses. First of all, this is not a complete table, so don't assume this is a complete table, but it tells us some very interesting things. So when you look at the DNA viruses on the table versus the RNA viruses on the table, what do we see? Which one is more common? RNA viruses. Okay. When we look at the DNA viruses, what is the most common form of the viruses that infect humans? Double-stranded, which makes perfect sense because our cells don't do particularly well with single-stranded DNA. When we look at RNA-based viruses, what is the most common form of the RNA-based viruses? Single. And then if we actually spend a lot of time looking at whether they're plus or minus, we see that they're more often a plus, although the difference between plus and minus is less drastic than being single-stranded RNA. And we have a number of very important viruses that I'm going to sort of talk about to demonstrate different things to you today. So amongst the DNA viruses, you have the pox viruses, the pox viridae, and those are one of the most important genera are the orthopox viruses, which of course are smallpox. But then you also have this group called the herpes viridae, which is actually a very important group of viruses. It includes anywhere from, the simplex viruses, which is the herpes type 1, which can be fever blisters. It can be respiratory infections. It can be simply your cold sores that you get, a cold sore that you get. But you also have other groups that include the type 2, which cause genital infections, and of course are these types of viruses, of course, have been tied to what cancer? You get a vaccination. You can get a vaccination now before puberty for these. Cervical cancer, but it also causes things like penile cancer and esophageal cancers. So the vaccine is critical now for people, both boys and girls, to get, because it protects them against a variety, not all of the different types, but many of them. You also have within this type the chickenpox, Epstein-Barr, which causes infectious mononucleosis, and again has been tied in some individuals to the development of Burkitt's lymphoma. So there's in some individuals, the acquisition of a viral infection can lead to ultimately at later in life presenting as lymphoma. And what we see in these cases of these cancers is not everybody who has an infection gets it, but there is a genetic component to what probably within their own genome that leads to that, as well as epigenetic effects of their environment. And then you have a variety of other viruses. Sorry, I misspoke. It's the papilloma virus. You should have corrected me, which it causes cervical and penile cancers. But again, double-stranded DNA viruses. The adenovirus is an interesting one because, in fact, we'll talk about it as a way to genetically modify cells, but causes a lot of respiratory infections. The picornaviruses include the rhinovirus, which is the common cold, which has many different serotypes, and is why, even though one would say, well, it's not particularly a bad infection. It does result in a lot of if you want hours at work in terms of people having to take time off, and you'd think that given its infectious nature, they might have attempted to create a vaccine. Anybody know, and I did actually mention why we can't actually seem to develop a vaccine to the rhinovirus? What might be the reason? There's too many variations. So generally speaking, if you get one rhinovirus infection, you'll be protected for that year. And if you do get another rhinovirus infection, it'll be from a different rhinovirus. And then you have the hepatinoviruses, which cause hepatitis B. We'll talk about that virus. And I only mentioned the parvovirus because the parvoviridae, because this is a very important, more of an important infection for dogs. So the dogs are now protected by a vaccine against parvovirus. Very important because many dogs, if they acquire this, especially as a puppy, will actually die from this particular viral infection. And of course, you have the very important picornaviruses, which include polio, as well as a different virus of the liver. So again, what you're starting to see with the various liver viruses, the viruses that infect liver and cause liver disease, don't even fall within the same category of genome. So we had the hepatinoviruses cause hepatitis B, and here we have epicornavirus, which is a single plus strand RNA virus that causes hepatitis A along with the rhinovirus. And then you have a series of them that you will have heard about right now amongst a couple of universities, especially Guelph. Neurovirus is causing great problems. It's very, very, very contagious. If somebody who lives with you gets norovirus, odds are you're going to get norovirus. It's a very unpleasant, fortunately short-lived gastrointestinal infection. And then you start to see this cluster here, which becomes very important that we'll talk about, the coronaviridae. And again, I made this point. You can get severe acute respiratory syndromes, but you can also just have the symptoms of a common cold. Within this, the retroviridae, of course, are the retroviruses. And then you have influenza following in this, which of course has this ability to be segmented, and that's also true for the retroviridae. And then you have the paramyxoviridae. So again, paramyxoviruses cause the common cold, but also within this group are the morbillivirus, which is measles, and the rubella virus, which is mumps. And of course, right now, what are we experiencing with measles? Throughout Ontario, southwest to substantially into Quebec, and all around the US, the reemergence of significant measles infections. Anybody have an idea of why they think this is happening? Because generally Canada has excellent coverage and has traditionally had excellent coverage of people getting vaccinated against this. With measles, you have to stay above, I think the number is around 95% of the population, but definitely over 90 has to be vaccinated or you will get outbreaks. What happened that may have caused this? That could cause it in older, but what happened that may have stopped the original first shot? Measles and the MMR vaccine is a two-shot virus.
Vaccine hesitancy.
That is playing a game too, but what happened in 2020? Yes, people have stopped getting boosters, but what happened in 2020 of March? COVID. And what happened in COVID? Everything was shut down, and small children didn't have access to getting their first shots in the time frame in which they could have gotten their first shot. So some of this is a product of the fact that children now are under-vaccinated because they did not get their shots in the normal pattern that they would have gotten them. So that's an important comment. And rabies, by the way, if you're interested, falls within this group. Any virus that I talk about again, you are responsible for. The viruses that I just mentioned in passing, you are not responsible for. So any virus that I talk about subsequently, you need to know about. But just because I talked about it or it was in this table, you do not have to know all those viruses. Yes, Andrew, do you have a question? Well, you have to know what they are. It's a multiple choice question. What do they cause? What are their symptoms? Or are they an RNA virus? Are they a DNA virus? I'm not going into that much detail about this. This is not a virology course. So this is just a reminder about what the 2003 SARS looked like, a helical capsid with an RNA. Again, we go back to our coronavirus table. It had a single RNA plus strand. It has the classic glycoproteins, and that glycoprotein predominantly attaches, that spike protein attaches to respiratory epithelia. And if we then, and there you see an EM picture on the left as you look at it. And then we have SARS-CoV-2, again showing you the helical capsid with the RNA within that helix. And you can see the studied important proteins that are on this surface, including the spike glycoprotein. And as I pointed out in the last lecture as well, it has this M protein, which we'll see again in some viruses that are part of the viral encoded proteins that help support the structure of the envelope. And again, so from a point of view of its structure, very similar to SARS, the original SARS, and also to MERS that I talked about last week. So remembering that organization of the viral genome can vary. We can have single-strand RNA or DNA. Others have different arrangements. Classically, the influenza virus, which has eight single-stranded RNA segments, which we'll use our influenza to really demonstrate the concept of antigenic shift in viruses. The viruses that have segments have the ability to do things that viruses that don't have segments can't. And so within this, then, we have variation of how and where within a host cell the viral genome is going to be replicated. Okay, so it could be replicated in the cytoplasm, it could be replicated in the nucleus, sometimes even into the endoplasmic reticulum, which of course is contiguous with the nuclear membrane. So based on everything we talked about this week and what I reviewed for you at the beginning, what we reviewed, the viral genome refers to the type of nucleic acid molecule found in the virion. Is that correct? So the viral genome is the type of nucleic acid that's in the virion itself. Virion means the viral particle. So yes, refers to the type of nucleic acid that is packaged in the capsid after replication. Is that true? Does all genetic material get packaged into the capsid first? Yes, all viruses have a capsid. Refers to the type of nucleic acid present in intermediates of viral infection. So a plus strand has to be converted to a minus strand in order to make more plus strand genome. Would that be part of the virion? No. So any intermediates are not part of the virion. We'll meet this again when we talk about the retroviruses who go through a DNA intermediate step. So what's the answer? Both A and B are correct. By the way, these are questions that when I give you questions in class, these are questions that have been and can reappear in so the forms that I'm giving you are the kinds you're going to see in your actual midterm and final. So when we look at a generalized view of how viral replication takes place, if we just tried to summarize it, yes, there are some variations that may occur with different organisms. We first must have an adsorption or attachment of the virus to the host cell. In this case, it's just showing you a naked virus. The genetic material must enter the cell. We will talk about the various ways in which that may take place. It's not just an injection as shown here. There's also forms of endocytosis that takes place, and we'll do that. But in general, somehow that genetic material has to get into the host cell, and then that genetic material has to be replicated, and then that genetic material has to be transcribed into RNA so that the viral proteins that are encoded in the genome of the virus are actually replicated. So at a minimum, the proteins that need to be made are the capsid proteins and the envelope proteins, and the proteins of the virus that get embedded in the envelope. But as we have variations in the way in which replication of the genome takes place, many times that virus is going to need support from specific viral proteins. So the retroviruses have to have support because our cells don't have the right polymerases to do what needs to be done. So if the type of replication that has to take place is different from classic DNA synthesis, then they have to bring in accessory proteins. And generally, they bring in both the gene encoding that accessory protein, and they package some accessory protein as well, so it can immediately be used. Okay? So we will talk a bit about that. And then, of course, the cool thing about all viruses is they self-assemble. They'll self-assemble in the host cell. You can self-assemble a virus in a tube. If you put all the various components of a virus in a tube, they can self-assemble without being in a host cell. Okay, so they have the ability to self-assemble, and then they have to be released in some manner. They could lyse the cell, as shown here, and that's because this is a naked virus, and really for almost all naked viruses, the only way they can get out is to actually basically lyse the cells, whereas the envelope viruses take on the membranes from the host cell, and they usually bud out of the host cell. So they actually don't cause complete lysis of the host cell that they've just infected. So viral attachment. It's not a random event. It's the basis on which viruses gain entry. It becomes critical to understand what those surface proteins on the virus, what they're attaching to. And it's going to be a specific viral component to a specific surface component on the host cell. So the receptor depends on the virus. So different viruses have different host receptors, and different viruses have different surface molecules that allow for attachment to cells. So what you might sit there and say, well, this is easy then. What do? We just get rid of the surface components on the host cells that the virus uses to attach, and then we never get a viral infection. Theoretically should work, right? But it can't, because the virus uses essential proteins on the host to actually attach. So you can't remove the host cell proteins because the host cell would not be able to survive. So a cell lacks the receptor. It cannot normally be the host. So in general, if I have 3 different cell types there, and one cell type has the receptor for the virus, the other 2 don't, those other 2 cell types will never be infected by that virus. Hence why we see the kinds of symptoms we get with different viral infections. A gastrointestinal virus is attaching to surface molecules on cells that are in the gut. A respiratory infection is attaching in the respiratory system. In viruses that have a more pleiotropic set of symptoms, such as, for example, measles or polio, then in fact what we see is they can attach to different regions of the cell, not just one. And I pointed that out to you about COVID. Most of the signals of having COVID, which is the actual infection, were respiratory. But sometimes they were gastrointestinal. And that was because the spike protein on the SARS-CoV-2 has the ability to also attach, not as well, but has the ability to attach to some gut epithelia. So, but remember, things that are in your gut have got into your body differently than viruses that infect your respiratory system. So you breathe in the viruses that are in your respiratory system. How do viruses get in your gut? You ingest them. Okay. All right. So they're always on the surface of the cell. So they always play an important role in the host cell. And so if it's removed, the virus may not attach, but it would be very detrimental to the host cell surviving. So that makes it more difficult to deal with the infection. So I'm going to use some examples, and I'm going to use the bacteria to demonstrate some of this. So if we talk about E. coli, bacteriophage lambda or bacteriophage T4 are both viruses that infect E. coli. And one of the viruses, bacteriophage lambda, is actually what you will be doing the phage assay with in the lab next week. So you're doing 3 experiments next week, as well as doing streak plating. You're doing a phage assay, you're doing conjugation, which we'll talk about in the coming lecture, and you're doing a chemical transformation of E. coli. So bacteriophage lambda binds to what's called the LAMB protein on the surface. It is actually a transport protein for the uptake of sugars. So again, E. coli can't lose that surface protein, because if it does, it can't uptake sugars. So the virus is using a very important surface protein in order to infect E. coli. So if it was missing, yes, lambda could not infect it, but it would lose the ability to use the variety of sugars it can use. So it would only be able to use a couple of sugars, which therefore would have a significant effect on its ability to survive. And one of the main sugars it would not be able to use is maltose. So if you want to make E. coli put out more receptors for maltose, what do you think you do? If you give E. coli glucose, it's not going to need a maltose receptor, so it's probably going to make fewer receptors on its surface for glucose. So what do you think would happen if you fed it lots of maltose and no glucose? It would make more receptors for they would make more LAMB receptors. So in the lab, if you pay attention, you're going to have the buffer that you have the phage in has maltose in the buffer to help those E. coli actually make more receptors, so you'll see a better infection with the phage. There are other bacteriophages like R17, M13, F1, FD. These are all variations of bacteriophages that affect E. coli. And in this case, these viruses all have the ability to bind to the pili, which we'll talk about in much more detail. But these are appendages you find around the surface of E. coli. They are not used for motility for E. coli. The flagella do that. But they're used for attachment to surfaces. And they're important as adherence factors. And they also become important for DNA exchange between cells. For example, the bacterium B. subtilis, which you were working with the last 3 weeks, it actually has a bacteriophage called SPO, and it actually binds the flagella structure. So in other words, different components of a bacterial cell, just like different components of your body, can be used by different receptors to gain entry into these cells. So these are just some examples of this. So you can see on the top here, these are EM pictures. So here you see all the different viral particles. This is actually, there's a pilus in the center here, and you can see all the viral particles trying to gain access. Here you see a phage that's landed on a flagellum, wrapped its tail fibers around the flagella, and will inject its genetic material into the flagella, which will then make it into the cytoplasm of the bacterial cell. And here you see the classic attachment of T4. Lambda does the same thing, where it's attaching to the cell wall, and I pointed this out in a slide last week as well. What you can see is, so these are already heads of a virus that have lost its genetic material. This one has just lost its genetic material. If you look carefully, it's down here. These ones, you can see the injection of the viral DNA being done. And what you can see is that if we went back to the original landing, the tail would have been much longer. So there's a shrinking so that the injection can take place through the tail fibers. And we'll talk about the process of that in a little more detail in a couple of slides from now. So the capsid or the envelope have the spikes that interact with the receptor. They're absolutely required in order for those viruses to be able to attach to the appropriate host cell. So there's some really interesting examples of viral receptors in multicellular organisms that help us understand why you get the symptoms you do. So for example, the polio virus. It has a specific glycoprotein as its spike protein, and that glycoprotein that it interacts with an ICAM. So it interacts with an adhesion molecule that's actually located in the cells of the human nasopharynx, the gut, and the spinal cord. So polio has this ability to infect at a respiratory location, and subsequently, by the way, become latent and reemerge. So many people who did get polio and survived in their iron lungs, because that's what, if you look at pictures from the times of polio outbreaks, you'll see rows and rows and rows of children in iron lungs, because their lungs were not working. They survived it, and then many years later, what would happen is they would end up with an inability to walk. Their spinal cord was basically, the virus became latent and then could attach to their spinal cord or into their gut. Some people ended up with severe issues with gastrointestinal problems as well. For the common cold receptor, the receptor on the virus binds also to an ICAM. So ICAMs are commonplace. They're basically ICAM means intercellular adhesion molecule. They're always glycoproteins in mammalian cells, especially humans. They're almost always they almost always contain sialic acid as the glycosylation, okay? And in this case, the common cold is in the nasopharynx and respiratory epithelium. So that's why your common cold always is respiratory. In a common cold, you don't get the symptoms of the flu. You don't get high fevers generally. You don't get aches and pains. You have basically a problem with the way in which your respiratory system is functioning, which usually means runny nose, runny eyes, congestion, cough, sore throat. Okay. In the case of measles, the protein that it binds to is CD46, which is actually found in a number of tissues. So in the case of measles, you really do have much less tissue specificity than other viruses. It has the ability to affect a lot of different tissues. HIV, on the other hand, is very narrow. It binds to CD4 proteins found on the plasma membrane of CD4 plus leukocytes. So white blood cells are leukocytes that are actually presenting with CD4 on their surface. And hence why it manifests by affecting the ability of the white blood cells to function. The rabies virus, okay, for those of you who have ever read about rabies infections, really the animals and the people that have rabies behave as if they're mad because their brain is being affected by the virus. And so it actually binds to the acetylcholine receptors in the neuronal plasma membrane. Okay. Fortunately, there is a vaccination against rabies for both animals. It's a little harder to vaccinate animals in the wild, but they do attempt to do that by baiting food with the vaccine. But for humans, if you do receive a bite from what they might think is a rabid animal, you go through a number of days, unfortunately, of very painful shots in order to be protected against that infection. The SARS and the SARS-CoV-2, it was the ACE2, which is the angiotensin converting enzyme. It's a blood pressure regulation protein that will cause leaky lung tissue, which was why it caused severe pneumonias and the symptoms that it did. But there is also receptors in your gut that are ACE2 in much smaller number, but the virus can still bind. In influenza, it's the hemagglutinin that contains a receptor binding site for sialic acid residues on the host cell surface, and again, more pleiotropic in where it binds. So host range is most greatly impacted by the presence of the specific receptor. Obviously, if those receptors are only found in humans, then only humans can be infected by that virus. If there are receptors that are commonly found in a variety of organisms, then you have the ability for that virus to infect more animals. Generally, however, the infection, as you see by the list, is likely to fall for humans within the mammalian population of animals. And remember, no virus can cross kingdoms. So if somebody says, oh, I don't like genetically modified foods because they use a plant virus to do X, well, that's just silly because the plant virus can do absolutely nothing to you. Okay? And for us, by the way, who knows one of the most important food stuff that we produce? It's soybeans. And they're all genetically modified. And they're perfectly fine. Because what is genetic modification doing? It's allowing you to grow the organism, the plant, in conditions that might have been detrimental to the wild type plant, but now you allow them to grow under conditions that are much more broad. And I'll talk about that at the end of the lecture. Tissue specificity can also become very important. And this became important for our understanding of thinking that viruses were a type of gene therapy we could use to treat certain diseases, because we could use the viruses to direct, let's say, a drug or a therapeutic gene to very specific tissue. And this was around for a very long time, but failed miserably multiple times. We now have some very good gene therapies, but still not as many gene therapies as everybody thought we would have, and there's a reason for that. Is because sometimes viruses that you have made innocuous to deliver your cargo can talk to the latent viruses that are in your body and reactivate because of the ability to exchange genetic material. And that happened more than several times in the original attempts in gene therapy. And the problem is they can't know all the viruses that you've been exposed to. So there are some limitations to the ability to use. That's why the viruses that are used are very controlled and go through very substantial clinical trials before they're ever used. All right, so what is involved when we penetrate into a host? So this is an example using the T4. Lambda would be the same. You've got the landing of the virus. And in this case, the genetic material is going to be injected, so the virus lands. It lands, the tail fibers attach. Eventually what happens is that tail shrinks, and the tail fibers actually create a hole here. And what ends up happening is there's a tail that then gets injected. The tail actually moves in through so this is a gram-negative cell. So we're looking at a gram-negative cell right now because T4 infects E. coli. So this is the outer membrane, and the tail moves in and comes in contact with the plasma membrane. This is the peptidoglycan, by the way, and we'll talk about that in much more detail. And that genetic material, which is double-stranded DNA, is actually injected into the cytoplasm of the bacterium. But the problem is peptidoglycan sits here, and you have to destroy what's left of the peptidoglycan to get through the plasma membrane, as well as there's some other enzymes that are involved. So there is actually an enzyme that's made by this virus as well. It's carried in its genetic material, and it carries in its head, which is lysozyme. Lysozyme breaks down peptidoglycan. Okay, so it actually encodes a lysozyme that actually you make in your tears, in your tissue, and on their skin surface to actually break down peptidoglycan. Okay, and you'll also do a lab on lysozyme. So the genetic material is completely injected into the cytoplasm. So that's an injection process. There are naked viruses that infect humans that will also just inject the genome into the host cell. So what has to happen then in order for the injection to take place or the entry of that virus to take place, it needs to adsorb to the surface, it needs to penetrate, and if it has a capsid that's entered the host or an envelope and a capsid that's entered the host, everything has to be uncoated in order for the genetic material to be released. So we need that initial contact binding to the specific receptor. We then need the induction of a number of processes. So again, as the previous slide shows, in some cases only the viral genome will enter. This is common amongst bacterial viruses, but it's actually rare in animal viruses to use that mechanism. Generally, the capsid containing the genome or the entire envelope virus actually enters the host cell and then becomes decoded. That's the most common way for most animal viruses. So there are a few naked viruses that will be endocytosed, and such as the adenovirus. And so again, there's an attachment of the capsid's spikes to the appropriate location on the plasma membrane, and you get an endocytotic event. And often this is going to take place in a very specific place that you will have already learned about in your cell biology and probably mentioned in a few other courses, often happening at clathrin-coated pits. So at clathrin-coated pits, you have a clathrin-mediated endocytosis. The virus is enclosed and then finally starts to be uncoated. This can happen, by the way, with both naked viruses and envelope viruses. There are certain envelope viruses that will also go through the process at clathrin-coated pits. But there's also other routes. So we can have receptor-mediated. We can also have caveolin-mediated, which is another form of endocytosis at specific places in the membrane where caveolin is found. We can have basic endocytosis, which means it doesn't have to occur where clathrin or caveolin is present. Or we can have fusion and delivery, which is shown here. So in this case, here's the spikes. This is the envelope. Here's your capsid with your genetic material. What's happening is that actually the envelope is fusing with the plasma membrane, and then the capsid containing the genetic material enters the host cell. So you have a variety of ways of entry, various types of endocytosis, as well as fusion of a membrane, the fusion of a membrane to the plasma membrane to deliver the host. Or, as in the case of the bacteriophage, just injection of the genetic material, and basically the capsid stays outside the cell. So if we look at this in a little more detail, this just demonstrates the various forms. So here we have, and again, you've learned about this type of endocytosis in definitely in your cell biology. So here's a caveolin coated pit where you're seeing, in this case, a naked virus being brought in, and then it fuses to create a particular type of phagosome or an endosome called a cavosome. And eventually, there can be activity here, or it can be delivered also to an early endosome. Same process with the clathrin-coated pits. It occurs there. The vesicle becomes, the clathrin gets removed, and then it binds to an early endosome, or just basically basic endocytosis, not at a particular receptor, enters the early endosome. Or it can be what's called a bulk phase endocytosis that brings it in, or just, and it can also be a phagocytotic event. For cells that are capable of carrying out phagocytosis, they can bring in viruses, and these viruses then are in a phagosome, and the lysosome will fuse with the phagosome and decoat the virus, and then the virus genome will escape and be able to be replicated. So basically, it's hijacking the various ways in which cells use to bring things into the cell. Types of endocytotic events, fusion events, as well as phagocytosis. All right, so we can use some examples of this. For example, the entry of HIV and influenza. So what you have is two different entries being shown on this slide to demonstrate this. So here we have HIV here. Here's your receptors on the virus. So the first thing it does is interacts with CD4, and then there's a second player in this interaction. And you can see that in the case of HIV, it's a fusion, and then the capsid with the genetic material is, it moves into the host cell, and then the capsid gets basically gets broken down by the mechanisms in the cell. And now we have the genetic material in the cell, and of course the first thing, because this is an RNA virus, it undergoes a reverse transcription. So can our cells carry out a reverse transcription? No, so obviously the reverse transcriptase and the accessory proteins needed for this virus are brought in on the genome, and also the proteins themselves are attached to the genetic material to support this. And then that double-stranded DNA after the process of reverse transcription and making a double-stranded DNA enters the nucleus where all the processes take place. And we're not going to talk about how it moves back out right now. Influenza, you can see the hemagglutinins. This is the HA. There's also neuraminidase receptors on the influenza. The neuraminidase receptors presumably help on both the entry and the leaving, so the removing of sialic acid residues on surface proteins of the host. In this case, you can see that this is happening at a coated pit where you form an endosome. So you have the membrane here. Here's the capsid with the genetic material. Here's your endosome. Eventually, this becomes a very low pH, which allows basically the removal of the envelope and the genetic material being released, which will then enter the nucleus to be replicated. And if you go back to your table, so if you go back to the table, you look at what kind of virus influenza is, and what kind of virus is influenza. And a plus or a minus? And it's a minus. So in other words, the first thing that has to happen is the minus RNA has to be converted to a plus RNA. Then the plus RNA will be used to make the protease, and then the plus RNA is used to make the minus change, gotta change the battery for the recording. All right. Now, let me stop my lecture. Okay. Okay, so based on our conversation about how animal viruses actually enter cells, a kind of multiple choice question I could ask you is, A, animal viruses can enter the host cell by receptor-mediated endocytosis? Is that correct? Hens, yes? Yes. Can enter the host by just plain endocytosis? Yes. Can enter the host by fusion to the host plasma membrane? Yes. So therefore, all of the above are correct. And again, this is a question that has occurred on exams before. And to be honest, just so you know, I am renowned for putting the same multiple choice questions sometimes that I give you in class right back on your midterm, where I discover you still can't answer the questions. All right. Okay, so we've got the virus attached. We've got shown how some viruses enter as examples to make the point. Now we have to go through replication, packaging, and exit from the host. So in the case of most viruses, the nucleic acid genome is released from the nucleocapsid, meaning the genetic material is first released from the capsid that surrounds the genetic material, so that's called the nucleocapsid. Then the viral genome has to be replicated. There is an exception to that, and that is that the plus strand RNA can immediately be utilized to make early proteins. Because remember, the plus RNA of a virus is in the same form as a messenger RNA in a host cell, so it immediately can be used to make some proteins. Meanwhile, the plus strand would have to be converted to a minus strand to replicate more plus strand for packaging of the genome. The viral genome, once that has been replicated in multiple copies, is going to have to be transcribed to generate the messenger RNA to make the proteins. And eventually, the copies of the viral genome are going to have to be packaged into new capsids to give those nucleocapsids, and then either exit the cell, or if it has to have an envelope, it needs to then interact with an envelope. It could be the nuclear membrane, it could be the ER, or it could be the plasma membrane that actually provides the envelope, the membranous material for the actual envelope. And then that virus can leave and infect more cells. So therefore, if it's a double-stranded DNA molecule, it'll enter the nucleus, it will be replicated, the messenger RNA will leave the nucleus, it will be translated into proteins, the virus will be packaged. If it's a RNA or a single-stranded DNA, there's usually going to be some kind of accessory proteins that are going to be needed because the ability to make a minus strand from a plus strand or a plus strand from a minus strand is not, the machinery is not in most host cells. So there is accessory proteins that are going to have to come in, and they will be encoded by the genome of the virus. And as I said, they often attach some of the protein they made when they package the capsid, so that when that genetic material enters, they already have pre-made some of the protein that they need to carry out their replication. So the double-stranded DNA use all host cells, but ultimately, even those that need accessory proteins are almost completely dependent on host machinery to make their proteins and to package to carry out the packaging process. So again, there's all sorts of variations of how different viruses do that. So we're really, in this course, I'm trying to give you a general overview of how this actually takes place. The key point is they're hijacking the process of the host cells to provide them with the precursors they need, the nucleotides for nucleic acid synthesis, ribosomes and amino acids, for example, to form the capsid, and also for the proteins that get embedded in encapsulated viruses. With very few exceptions, viruses that have DNA genomes replicate in the nucleus, and the viral genome is actually packaged in the nucleus, generally speaking, for animal viruses. So what does this tell us? That the messenger RNA leaves the nucleus, makes the proteins that are needed, and then the proteins are moved back into the nucleus to actually package the virus. Okay. The packaged capsid ultimately leaves the host cell and can infect other cells. If it's an envelope virus, however, they either bud from the host plasma membrane, as I mentioned earlier, which occurs with the influenza virus, although the influenza virus can occasionally bud from other locations, from the nucleus in the case of the herpes virus, which is a double-stranded DNA virus, or bud from the endoplasmic reticulum or Golgi, because these are membranous structures that are either contiguous in the case of the ER, or they can get into the vesicles of the ER that end up actually moving to the Golgi. All right. Once the mature viral particles are released from the host plasma membrane, that would be a process of exocytosis. So anything that leaves via budding off the plasma membrane is an exocytotic event, making use of exocytosis in the cells. The membrane from which the virus buds are altered, often to include viral envelope proteins, so therefore the viruses actually are surrounded by the important proteins. However, this is an important thing to remember. So remember, the host membrane has its own proteins. So in order for the virus to have its proper surface molecules, the host proteins have to be excluded from that area, and then the viral proteins have, have to be put into the membrane. This is not always accurate, which means sometimes either viral proteins are left behind in the host cell, or host cells end up in the viral protein packaging, which then can cause issues around autoimmune responses if, in fact, a host protein has now been packaged with viral proteins on the surface of a virus. So that adds some variation. Generally, the virus is very good at this, but sometimes mistakes get made. Okay, so that's an important thing to remember. Naked viruses usually leave by lysing the host cell. So if we use, again, a bacteriophage as an example of the important steps, what we see here is, again, this is T4. It lands. Its genetic material gets injected, okay? And now it's going to make use of the proteins that are actually in the bacterial cell, so nucleases, DNA polymerases, and sigma factors to actually copy the phage DNA. So the infection takes place. And what's going to have to happen is there is some early mRNA process, because sometimes they need some new sigma factors, so they have to make some specific proteins that are unique to their process. In the middle range of the infection, the phage's DNA is being replicated, and then you get some more mRNA being made. And then following DNA replication, the last set of mRNA is made. And you also have production over the time of early proteins, mid proteins, and late proteins. Note the time here. This is important for your experiment. In 25 minutes, this virus has entered, done all of its business, and is ready to leave that cell and kill that bacterial cell by lysis. So if you are not paying attention to your time, and you go past approximately 15 to 20 minutes, you risk missing the burst of release of phage for your experiment. So the timing is actually quite critical when you plate onto the plates. That's why you're asked to complete that in 15 minutes. because this whole process takes very little time to make the phage head proteins, the tails, etc., the mature phage particle, and again, make sure there's T4 lysozyme production to package into the cell because the phage, because now they have to lyse out of the cell too, which means they have to break the cell wall. And to do that, to break down the peptidoglycan, they make use of, again, T4 lysozyme production. So the interesting thing, and the phage are very interesting to use as examples because they demonstrate the variations in which viruses can exist in a cell. So you're going to do an experiment on the lytic phase of Lambda in lab next week. But there's lytic phases, and by definition, lytic means it's going to cause the lysis of the bacterial cell, so the phage is going to multiply, it's going to make hundreds upon hundreds of phage to be released to go ahead and infect another cell. But there's also what's called a lysogenic phase. So what you're going to do in lab is do a classic lytic phase assay. You're going to take, you're going to add phage to the bacterial cell. You'll allow that phage to attach, give it time to infect, and then what you're going to do is you're going to add it to a molten top agar. So this is a liquid top agar that when you pour it onto a plate will solidify. It's kept at 50 degrees for you so that you don't kill the bacteria, but rather allow the bacteria to survive. So you pour that on top of a nutrient plate, which now has the viral mixture that's infected the E. coli. And what's going to happen is that top agar, as it's called, is going to spread and solidify, and it's there that the phage, you'll start to see the plaques, which will represent a phage infection. Remember, there were also bacteria here, so any bacteria that does not get infected will grow as a colony. So really what you end up with, because you have so much bacteria there, you have a lawn of E. coli with these clear zones that are round that are called plaques, which represent an infection of an E. coli cell. So if timing is correct, you can count the number of plaques. So if you look carefully, you can see them on this slide. And this is also, just turn this off for a minute. You can see the light areas. You'll also ultimately, there's also a picture in your manual. That's what you're going to count the following week. And from the count, you can determine the original phage suspension concentration. Because by definition, just as a single colony was given rise to by a single bacterial cell, a single plaque was given rise to by an infection of a single E. coli cell. Then there were multiple rounds on the plate after you plated, so that you actually can see the plaque, which will have millions of phage particles. So that's what you'll do next week, and that's actually your first formal lab report, by the way. But the interesting thing is the lytic pathway is an induction and ultimately release of the virus from the cells, killing the bacterial cell. But there's also what's called a lysogenic phase. So under certain conditions, when that viral DNA is injected, instead of causing a lytic phase, it actually results in that phage being integrated into the host chromosome. Really, this is what retroviruses do. They integrate into the host chromosome. Any virus that can integrate into a host chromosome is not a safe virus, because when it integrates in the host chromosome, what is it doing to the DNA? It's disrupting it. So it can cause damage to the DNA, and hence viruses that can do this have been tied to a possibility to transform those cells into cancerous cells. So at this point, it's a prophase. It's sitting quiescently. It's not doing anything. And it'll replicate with the host chromosome. So if there's any interesting genes in that virus, now the host has them. So maybe this virus has collected antibiotic resistance genes over its time. So this is one of the ways in which viruses collect antibiotic resistances. However, the interesting thing is in the case of the bacteriophage, so cell division takes place, so You keep passing that lysogenic virus onto the progeny, but at some point there can be an induction, usually a stress, that causes the phage to actually make its way back out of the genetic material, and it enters back into a lytic phase and kills the cell. But meanwhile, it's been passed to multiple bacterial cells and given those bacterial cells extra genetic material that may allow them to do things that they couldn't do before. And this will become important when we talk about horizontal gene transfer. So if we look at just an overview of the phage life cycle and classification, we can see that there are different types of phage. They're not all double-stranded DNA viruses. A large number of them are, or single-stranded, but there are some RNA viruses as well. And many of these viruses have both a lytic or lysis cycle and a lysogenic cycle, where they can live in the host genetic material and sit quietly for long periods of time and basically pass that genetic material on every time that cell divides. And what we know about our gut is that our gut is full of bacteriophages, which has become very important in trying to understand the gut microbiome, because those phages have the ability to target certain bacteria. And what phage is in your gut may actually affect the kinds of bacteria that you may actually be harboring. And so this also ties to the fact that when you get a fecal transplant, which really is somebody, if you have a severe irritable bowel syndrome or other types of syndromes, when they inject a healthy individual's basically feces up your rear end, they're not just injecting the bacteria in the living cells, they're also injecting the bacteriophage that were in that person's gut. And so this is a very important area of study at this point in time as well. And if you're interested, this is actually from a 2024 review in Frontiers in Microbiology. So just going to run through a couple more examples of the process using a double-stranded DNA virus. The key point is, okay, so it's a double-stranded DNA virus. It's the herpes simplex, which has an envelope. It undergoes a decoding at the membrane. That genetic material enters the nucleus. It undergoes replication. mRNA is also made. That mRNA here exits, makes the proteins. So the proteins of the capsid, proteins that will be embedded in the envelope, as well as any other important proteins. And then those proteins that have to make the capsid, they actually enter back into the nucleus through the import process of the nucleus, and you actually get the capsid surrounding the nucleic material. And then what happens is that the spikes actually get embedded in the nuclear membrane. So they don't actually enter back into the nucleus, but rather they actually get placed in the, they actually get incorporated into the nuclear membrane, and then that virus pinches off from the nuclear membrane, and then it does have to disrupt the host membrane to actually be released because it already contains a membrane. Okay, so that's a classic way, for example, for a double-stranded virus to work. And no, I'm not going to give you about 5 different combinations and say pick the order that the virus uses. I want you to understand that the herpes virus is a double-stranded DNA virus. Where is it packaged? Very simple information. Okay, this is not a viral course. I'm not going to ask you to tease apart every step that occurs during the process of these viruses. If we use SARS-CoV-2 as an example, that's shown here. And what we see is, here again, it binds to the receptor. It's going to move in by a receptor-mediated process. The genetic material is released. The ribosomes engage because this is a single-stranded plus strand. It can immediately start to make the proteins that are needed. Meanwhile, the replicase that was brought in at the same time is going to convert the plus RNA to a minus RNA, and that's what's going on here to make the genome. So you end up ultimately able to make the various proteins you need, and then you end up starting the process of embedding those membrane proteins that are going to be in the envelope. And what you see in the case of SARS-CoV-2 is the injection is actually in the endoplasmic reticulum, or it can also take place in the ER-Golgi intermediate. And ultimately, the genetic material ends up inside this package and ends up being exocytosed. So another classic way in which this can take place. And then finally, the retroviruses, to finish off various classifications. Again, in the retroviruses, there is a fusion of the membranes. The capsid enters, it decoats. You have the viral RNA. You notice that the reverse transcriptase not only is encoded in the viral genome, but also comes in as a protein. You get reverse transcription to get your double-stranded DNA. Your double-stranded DNA enters the nucleus. It becomes a provirus inside the nucleus. And basically, the mRNA gets made, as well as the viral genetic material gets made. And then it moves out into the cytosol where the proteins get made. And ultimately, there's vesicles that are made where you put in the budding. It buds out of the plasma membrane. So again, in this case then, when this forms this structure, it could, in the way in which this gets incorporated as a provirus, it can disrupt the genetic material. And as such, this is why retroviruses have been tied to certain genetic mutations and also to individuals who may well present with certain types of cancer. And this is just the EM picture of that. So basically, we've reached a point where I'm just going to talk a bit about some of the characteristics of some of these viruses. So it's about, we'll use that clock, it's about 14 minutes after, so come back at about 24 minutes after. We are doing 2 questions today. Okay, so we are doing in-class exercise today. So when you come back, we'll continue with the slides, and then I think there's a in-class about 2 slides in. Okay, so come back in about 8 to 10 minutes. The RNA genome, and here you see, in fact, not the virus, but the viral DNA, or viral RNA. It's a genome entry.
So it enters the nucleus.
If it enters the exit, if the genome is exiting the nucleus, then it's going to exit either by budding or by destroying the nucleus.
Okay, so the proteins, they exit. I was just trying to remember what happened. They enter the nucleus and the proteins go out.
Well,
Transcribe.
yes, the proteins have to be, the mRNA has to be made. The mRNA goes to the cytosol. The proteins are made. The proteins if the capsid has to be made, the proteins enter back into the nucleus by the importing process. They make the capsid, then the virus leaves. Okay? So it's using the normal cell machinery of the cell. So we make proteins in the cytosol, right? So that's where the viral proteins are going to be made too. Regardless of where the genetic material is, the proteins are always going to be made in the cytosol.
For the in-class assignment, like, do you have to talk up to it, or like we are already there?
No. Okay, we'll get started. Okay, so this is just a cartoon of the retrovirus structure to make a point. So we've been talking all along about the need to bring accessory proteins if, in fact, those proteins don't exist in the host. So this is just demonstrating that. Okay, so here you have the RNA genome, and here you see, in fact, that part of the packaging into the capsid includes that accessory protein, which in this case is both the reverse transcriptase, the integrase, and the protease. So remember, there are 3 enzymes that become critical to actually carry out an infection with a retrovirus. Those 3 proteins are needed. They're actually contained in the genome of the virus, the GAG, the POL, and the envelope proteins. All of those are encoded, as are so the envelope is the protein that goes into the envelope. These are the ones that are going to be important for actually the packaging. And of course, the treatment of HIV involves a cocktail that will prevent the ability of these proteins actually to be made and function. So the last step we have to do is understand how the virus leaves. This is just demonstrating what would happen at a plasma membrane where you have the genetic materials encoded in the capsid, and now we're going to form an envelope from the plasma membrane. So you can see that, in fact, the envelope proteins have been embedded into the plasma membrane of the cell. Eventually, there's an exocytotic event, and this virus then gets released. Now, there's some other proteins that become important in this packaging process. And that's demonstrated with, this is influenza. So you can see again, here is the helical genome in segments. And what you see is the first thing that surrounds this in this packaging is the M protein, or the matrix protein, that's going to be critical in the envelope. It interacts with the spikes that have been put into the host membrane, both the neuraminidase, which is a popsicle shape, and the hemagglutinin is a bat shape. And what you see to understand is when these proteins have been placed into the plasma membrane, there are still host membrane proteins present. So they're what's in the green. So you can still see they're there. There's host proteins there. And eventually what happens is those proteins get excluded, and there's a pinching off. But you can also see that very often the viral proteins are left behind in the host cell that has been severely damaged by the infection. And occasionally, some of the host proteins may actually end up going with the viral particle as well. All right. So this is our first thinking question. I'll give you your piece of paper. You get into groups of, if you want, anywhere from 3 to 5. You write your names neatly so I can read them. Please don't put your student number on them, only your name. And the question is, if an envelope virus was somehow released from a cell without budding, in other words, it does not have an envelope. So it's an envelope virus. It needs to have the envelope placed on that virus, but for some reason it gets out of a cell without getting its envelope. So it would not have an envelope. What effect would this have on the virulence of the virus? So that's the question. So if you understand what we've talked about, you can answer that in 1 to 2 sentences. So put yourself in groups and put up your hand for your piece of paper. Are you working here?
Thank you.
Paper? Paper? You need paper if you're going to do this. So the idea is you don't look it up, you use each other to answer the question. And it requires you, therefore, to have, can you pass that down? Conversations that revolve around your course material.
Thank you.
Can you pass one down for me? Whoops. I hate this room. I hate the way the stairs are configured.
Write our student number as well.
No, just your name that I can read. You need a piece? That's okay.
Thank you.
So you have a couple of minutes, 2 minutes to write your answer. Paper? Are you doing the assignment or are you working with him?
Yeah.
Okay, then it's fine.
okay?
That's fine, no problem. You can always use your own paper if you want to bring your own paper. You need paper or you're working together? Okay.
Thank you. And we put our group's name on
No, put all of your names on it.
Yeah.
Yeah. All your names need to go on the paper so I can give you credit for the exercise. Paper? Oh, sorry. There you go.
Sorry, I'm sorry. Sorry.
There you go. So you are working? Okay. Anybody else need paper? Working by yourself? Okay, well, maybe they'll let you join them. There's 3 of them behind you. Why don't you just talk to them? You don't want to do that?
No, I just said yes.
Okay. Okay, so literally it's a 1-sentence answer if you know what the answer is. Okay. Okay. Okay, 1 more minute. Okay, so what's the answer? If this virus doesn't have an envelope, what's the effect on the virulence of the virus? So theoretically, it's lost its virulence. It has no ability to enter another host cell, so it becomes a useless virus, theoretically. But what if it finds its way by some mechanism, it gets picked up and it ends up in a host cell? Is it still not dangerous, or is it now capable of being replicated in that cell?
It depends. If the virus is a positive RNA, like the COVID, they can immediately start turning its RNA to protein.
Okay, so if it did find its way into the cell, then you're saying it could still replicate its genome? Could it still make proteins? Yes. So it could still be packaged. So in other words, yes, it is true. Without the surface molecules, it should be a dead virus. But if by some cellular mechanism it ends up riding along with another virus and gets phagocytosed or enters in some way, it is still capable of carrying out all the processes that virus would have been able to carry out. Replication, making its spikes, and now properly making the envelope. So we need to think about, so that really demonstrates to you that the material for the purposes of replication and making those spike proteins is still going to be present in that genome. All right. So let's turn our attention a bit to keep that because there's one more to influenza to make a point. So influenza has that helical capsid. It has neuraminidase and hemagglutinin as its spike proteins, and of course it has its segmented genome. And it's the segmented genome that ultimately makes life miserable for us as it relates to to figuring out what the flu epidemics are going to be. So neuraminidase is a sialidase. It means it removes sialic acids from glycoproteins that could be found in the host receptor. It's really thought to help in the aid from the host cell, but also may help to alter mucus covering membranes for entry. Hemoglobin is really the spike that causes the host cell membrane to bulge inward to form the vesicle for receptor-mediated endocytosis. So both of these are spikes, and both of them can get altered, and both of them are encoded by regions of in different segments of this genome. So there are three types of influenza. Influenza A is responsible for pandemics. Remember, a pandemic is simply epidemics that are spread over large geographical areas. They're caused by variation in both hemagglutinin and neuraminidase. The word pandemic or epidemic does not signify severity of disease. It simply signifies a spread, okay? You could have a very small epidemic of a very serious virus, which could have more morbidity and mortality associated than a pandemic with a virus that has less morbidity and mortality attached to it. Influenza B generally cause localized outbreaks or epidemics, but have no clear subtypes. So they don't show variations in the hemagglutinin and the neuraminidase. Influenza type C causes very mild symptoms. There are no clear subtypes. You probably often don't even really, probably have a bit of a stuffy nose and you don't even know you have an influenza C infection. So really it's A and B that causes pandemics almost every winter. And really it's influenza A that is problematic in its ability to undergo major changes in the genetic structure of the virus because of the variations in the hemagglutinin and the neuraminidase. So there's two types of genetic alterations we can talk about for viruses. Antigenic shift versus antigenic drift. So antigenic shift is very distinct from antigenic drift. So antigenic drift is what happens in all genomes. It's a slow mode of genetic change of viruses over time. Antigenic shift, and this is a specific term usually used for influenza, so it's called viral shift or reassortment for other viruses that have segmented genomes, allows for two different influenza strains or two other viruses that have different strains to combine to form new subtypes, having mixtures of the surface antigens of the two original strains. And this becomes very possible because of the ability to exchange different segments of the genome, which you'll see in a minute. So you can think of antigenic drift as natural mutations that occur over time. This is what we just lived through with SARS-CoV-2. These were natural mutations that occurred over time. The virus was spreading at great rates, so therefore the mutation rate was very high. Antigenic drift occurs in all types of influenza, but only influenza A undergoes antigenic shift, because only influenza A has the opportunity for a major reorganization of its surface antigens. And influenza B and C only infects humans. A infects birds, humans, pigs. So the variations of hemagglutinin and neuraminidase results in viral subtypes. So if we go back to the original viral pandemic of 1918, which was an influenza type A of an H1N1 subtype. And actually, what people did is they actually went and they resurrected this genome, this virus from the cadavers of soldiers that had died during World War I to allow them to try and figure out why this was such a severe infection. Because people who suffered from this particular subtype of influenza literally drowned due to the fluid in their lungs. It was a very severe influenza subtype. And if we remember, this was at a time when people were returning from the war, so this virus was being spread all over the world. And actually, the second round of the infection, when they thought they had it under control, was much more problematic than the first round of infections that occurred with that particular influenza. So again, antigenic drifts, small change due to mutations. They're random point mutations. This can occur in hemagglutinins or the neuraminidase, which can affect the ability of a host to destroy a virus. But the antigenic shift results in major changes due to recombination between strains. So recombination of their genetic material. And this creates a different virus. And oftentimes, it can lead to a type A influenza that has never been seen before, which means there is nobody with any actual immunity to that particular virus. So this can lead to new highly virulent strains. So this is demonstrated here. So what you have at the top is drift, just to make the point. Okay, so here you can see here's the virus, and there's a mutation. See this red band, there's been a mutation, then there's a second mutation. But when we look at the structure of the virus, there's a little bit of change in the triangle here, which is the H antigen, but there has really been no change to the neuraminidase, and it's a minor change. So, you know, it might have a slight change in the way this virus attaches to its host, or the way in which the host, how easily it infects the host, but it's a minor genetic change which we would expect over time for any genetic material to undergo. Okay. Whereas in this case, what we have is a host cell in which we have two different virions. So here is an influenza virion from, let's say, a pig or a bird, and here's one from human, and it's sitting in a host cell. It could be sitting in a human, it could be sitting in a bird, it could be sitting in a pig. And what's going to happen is there's going to be resortment. So this is easy to look. The triangle and the popsicle are all red, and here you have a purple triangle and we'll call it a orange popsicle. They undergo genetic mutations in that they exchange segments of their genome. So now what we have is one segment from this virion joining 7 of the segments of the human influenza virus. We now have a different virus. And if we look at our surface molecules now, what we have is the red, this particular piece of genetic material encodes the H antigen, and now we have the H antigen from this virus, and we have the N antigen from this virus. So we've had a total reassortment to this virus, and this virus has gained one of the segments from this virus, and now it has, if we look at the combination, it now has, instead of having these two surface molecules, it actually has gained 2 completely different surface molecules. So this is what happens during influenza. This is what leads to the pandemics, this massive resortment of genetic material. And and the point is this can happen whether it's hemagglutinin or neuraminidase. So this just is a cartoon that demonstrates this. It says, you know, this is a chicken talking me. I feel great, honestly, just fine, not even a hint of a sniffle, never been better. But inside that bird is a bird virus. And then the pig gets infected with the bird virus, and it's already carrying a human virus. And in the pig, there's a total reassortment that can lead to, and the human virus is now, as you can see by the picture, no longer looks like the original human virus, but has undergone a reassortment with a combination of the virus from the influenza virus from the bird, and now you have a resorted virus that has different antigens. It might now be an H1N5. It could be an H1N2, different subtypes that can lead to a very substantial outbreak of human influenza. To make the point, this is what happened during COVID, which demonstrated genetic drift. So if we take March 2020 as our starting point, if we look prior to March 20, okay, you look at what the types of viruses that were present. Almost all the virus that was in North America, actually, if you look carefully, was of what was the subtype that was in China. But meanwhile, in Europe, there had already been a modification by genetic drift to the European form of COVID. And we are using European just to mean that mutation occurred in Europe, had nothing to do with Europe per se. But it did exist a tiny bit in China. And then by the end of March, you can see what has taken place. The original version, the version that came out of China, the genetic code that was there, has now been replaced almost exclusively by that which mutated in Europe. And now you see this in March. And this just shows you the ways in which it moved and how fast the variants actually moved. And you can see that in different places like North America did have some of the original subtype, but then quickly took on the other subtype. And so the point is, what was this change? Okay. So the global transition from the original position at 614 from an aspartate to a glycine, so basically a single base pair change, led to this form. So again, orange is the original that came out of the original genetic material that came out of China. B is the European variant. This was the first variant discussed. And now we see multiple variants. And we keep seeing those variants replace previous variants. But in reality, the original vaccines still work against the new variants. Not quite as well, but they all still work against the original variants because it's really single base pair changes that are occurring in various places, sometimes in the spike. And remember, you might say, well, are all changes good for the virus? No. There could be a mutation that ultimately leads to the spike not being able to attach to the host receptor anymore. Or the variation may lead to a greater ability to infect, but a poor ability to actually cause morbidity and mortality. So all changes to the genetic material are not positive for the virus, and viruses undergo this constantly. So the last thing I really want to really address in great detail today is the types of viral infections so we understand. So we have acute rapid onset viruses that last a very short time, like influenza. We have persistent infections that can last years. These can be chronic viral infections like hepatitis B or HIV, where at some points of the infection, where infection has taken place, there really are no symptoms yet. But over time, the symptoms emerge. And this is why you can have hepatitis B carriers. They're carrying the virus, but they have no symptoms and can therefore, infect and cause problems in food prep, et cetera. We can also have what's called a latent viral infection, where the virus stops producing and remains dormant for long periods of time and then becomes active. This is actually what happens with, for example, the herpes viridae, the herpes simplex virus that causes genital herpes and cold sores. The disease is contagious during an outbreak and is really incurable with present technology. In other words, we can't stop the virus from hiding. And so the cold sore, which is not a canker sore, by the way, emerges again because of where the virus hides. It hides around your trivaginal, your nerves here, and then reemerges when you're under stress. Same thing happens with Epstein-Barr viruses, which is the cause of mononucleosis and also causes chronic fatigue syndrome. And so this virus can hide and then reemerge and then disappear again, and then somebody can represent with the similar infections. And the reality is basically every single one of us has been exposed to Epstein-Barr virus and we never got sick. it is asymptomatic to us. But for some people, it becomes a real problem. And really, once you've been exposed, if it's latently present, it can reoccur at any time, generally during illness or stress, and can cause issues for people who have autoimmune diseases. And depending on some of the epigenetic and genetic makeups you have, it has been tied to Burkitt's lymphoma. Another example of this is the chickenpox virus. So that's varicella zoster. You have an acute infection, it goes away. But it hasn't gone away. It's just hiding. And at some point, it reemerges as shingles. Okay. So shingles is actually a reemergence of chickenpox. And actually, the reemergence of shingles is actually even a more severe infection for the people that get it, because it's severe nerve pain. And for some people, they can't even put clothes on. There's so much nerve pain from shingles. So shingles becomes the latent infection. Now, what we don't know is, depending on, yes, perhaps the majority of you sitting here have been vaccinated against chickenpox. But if the chickenpox vaccination was a live attenuated virus, there is no real indication of whether or not you are going to need a shingles vaccine. That remains a big mystery about the vaccinated core of people who received the chickenpox vaccine. Whether or not you are still going to have a shingles vaccine to protect you against a reemergence. And the literature does not address this. And how do I know this? Well, see, I'll give you a little aside. I didn't have chickenpox as a child. I should have. I'm of the age I was in rooms with lots of chickenpox, but I didn't get it. And so I had to go through chemotherapy. And so the concern of my neuropathies was they didn't want me to present with shingles at some point. So they wanted me to get the shingles vaccine. So they tested me. And I didn't have chickenpox any evidence of antibodies to chickenpox. So then the question became, do I really have to worry about shingles? But the decision was to vaccinate me against chickenpox. But then the question became, should I get vaccinated against shingles too? My theory was no, because why do shingles reemerge? Does anybody know? It has to do with your antibody titer. So if your antibody titer stays high, shingles will never emerge. But as you age, your titers to diseases that you've had, you've raised antibodies to, your titers drop. Okay? So theoretically, if I got my chickenpox vaccination at the age of 40, I really shouldn't have to worry about shingles. But the decision was I should have the shingles vaccination. But that was the crappy shingles vaccination. So then I just had to have the double shingles vaccination, because they actually don't know the answer to this. They don't know if the chickenpox vaccination will prevent you, because the vaccination doesn't stop you from getting a chickenpox virus. It stops you from, it does reduce the chance, but it doesn't stop you getting exposed to a chickenpox virus, which may well remain latent in your body. So that becomes the question that we still haven't answered about the need for getting a shingles vaccination. We'll see what happens in the next 15 years or so. So again, shingles has a slightly different name. It's called herpes zoster. So basically, for every 10 people who had chickenpox as a child, they will get shingles as an adult, and it's not always just late in life. There are people who, because of their immune system, present with shingles in their 20s. There's children who've had chickenpox and then gone through chemotherapy and then got presented with shingles as a young child. So this virus awakens, it causes severe sensitivity, it can infect your eye, it can occur in your eye, it can occur on one side of your body, it can occur in your mouth. It's not contagious, so you can't get shingles from somebody else. But if you haven't had the chickenpox vaccine and you haven't had chickenpox, you can get chickenpox from somebody who has shingles. So if you're looking after somebody who has shingles and you haven't been vaccinated and you never had chickenpox, you may get a surprise one day after a couple of days when you present with chickenpox. So acute viral infections really, the bar is where the viral load is, or the infection takes place. The red is where you get the symptoms, and the blue is the viral load. And the acute infection is easy to understand. You get it fast, the virus is gone. Influenza, smallpox, SARS, although now we have to ask what is going on with people who seem to suffer from long-term COVID symptoms after SARS-CoV-2. We really don't know why. You have chronic infections where you have the virus, the infection and the virus stays here. The viral load goes up, but you actually don't get the actual symptoms of the disease till later. And then you have latent infections that can have episodic emergence, like shingles, or you can have what sometimes is called slow, which is also sometimes latent, where you have an immediate infection, you get some symptoms, then the virus titer goes down, and then at some point the virus titers go back up, and then you get the real severe infection that will lead to, in the case of HIV, death if you haven't been taking the cocktail. So just to make a point about cold sores and where the virus hides, when you get the infection with herpes simplex virus, the virus then starts basically it hides, and it hides in the, as I said, through the trigeminal nerve, so it hides in your nerves, and that's why with cold sores it emerges where it does, because that's where the latent virus is hiding. And by the way, the emergence of this is usually a stress, like you have another infection. That's why it's called a cold sore, because often you get it right after you have a cold. You could get it from a very bad sunburn, where your body is stressed. All right, so now we have our second critical thinking question, which you can write your answer to. A latent virus that is incorporated into the eukaryotic host cell's chromosome is never induced. In other words, it never emerges from the host cell to be a free virus, unlike the ability of lysogenic viruses to reemerge. An example, the HIV virus could do this. Are such latent viruses considered safe? Write your answer and why. If it's sitting in your genome, not doing anything, is it safe? You're assuming it never becomes an infectious virus. It's just sitting in your genome. So, has everybody written an answer? Not yet. Okay, one more minute. Come on, your instinct. What is your instinct to say? What's that?
Can I ask you?
Sure.
You said HIV is it chronic or is it acute or is it latent? Because here you said it was chronic.
But it's often called slow chronic, because what happens is it emerges quickly, there's acute, and then it just disappears. Whereas, for example, let's say hepatitis, etc., here will keep a very high titer of the virus, but the symptoms appear later. Whereas HIV, even the viral titers go down and then come back up again.
Okay, thank you.
No problem. Okay, so what do you think? Is it safe? Why is it not safe?
Maybe under environment changes and under favorable environmental condition, it can reemerge.
Okay, yes, if you could impose that, but let's assume that can't happen. It's just sitting there in the genome. Let's assume it can never reemerge. Right, right. So the point is, any piece of genetic material that it gets placed into your genome, be it a transposon, be it a virus that is latent, can disrupt the genes that are present in your genome and cause mutations and can start the process of a transformation. It alone is unlikely to cause the cell to directly transform into a cancer cell, but it can start the process of creating mutations that lead to can lead to cancer. But even without that, if it went into this gene, is this gene functioning any longer? No, it's not. So you've lost function of something in your genetic material. Yep.
Isn't it true that there are cases of patients with low HIV, they have low cancer HIV, low cancer virus in their system, and it's all non-dangerous?
With HIV, the problem with HIV, which is the slow, is that it usually emerges quickly as an acute infection, and then you notice the virus disappears for a period of time and then reemerges. It's different than hepatitis, where the symptoms don't occur till later, but the viral load is still high. So yes, and the advantage of the treatment for HIV is that the cocktail basically keeps that viral load low and keeps it there, and in some people totally disappears. And even antibodies to the original outbreak of the original acute infection actually show that there's no evidence that the virus is present. But if you're not taking the cocktail, that's the problem with HIV. Symptoms disappears, and they can't find the viral load. And then suddenly the virus starts to replicate. Many years after you were infected. Okay? All right, we're going to stop there. I'll finish this next week and talk about the first few slides for the midterm. Remember, when's the midterm? The 12th. Hand in your sheets before you go, please, on the desk.